Hey, this is Dr. Arpin. Thanks for joining me on this first pre-laboratory discussion on this awesome experiment, hemoglobin drug synthesis and assessment. Today we're going to be focusing on the synthesis portion of the experiment. So the whole goal of the experiment is to identify drugs that bind the active site of hemoglobin. And you're going to accomplish this goal by actually preparing different drugs. You're going to test them for their binding activity, and then you're going to identify the best binders as potential drugs for hemoglobin. And so by accomplishing the entire experiment, I hope you will gain exposure to the field of medicinal chemistry. You'll definitely get to practice drug synthesis, and you'll get to execute and hopefully understand a protein binding assay. And finally, you'll get to analyze a larger set of data to identify those effective drugs. Thinking big picture, the experiment is comprised of three main parts. First up is the synthesis, where you actually make one of the drugs belonging to one of these four scaffolds. You'll then test your drug in the assay portion of the experiment. So this is where you'll be performing the protein binding assay. And the third part is analysis. This is where you'll examine the efficacy of your drug and compare it to other drugs to discern those most effective binders of the hemoglobin protein. Since we are making drugs, we should probably discuss how drugs work. And the bottom line there is that every single process in your body takes place through a very, very steppy and complex chemical pathway with all sorts of different proteins and different signaling compounds that get passed on from one to another. And whenever these signaling cascades or processes have some sort of abnormality, as in they're turned on too much or they're not active enough, whenever we have some issue there, this leads to disease. And so what drugs can do is they can go in and have a specific effect on those pathways. So for example, what's shown here is this STAT3 pathway that is overactive in cancer. This is turned on all the time. And so what we can do is imagine building a drug that actually prevents this cascade from happening. And since the cancer cells rely on this overactive signaling cascade, our drug will be selective and kill cancer cells because we are inhibiting it from taking place. So how are drugs normally discovered? It can take on average between 10 to 15 years to identify a drug and then get it to the market, which translates to possibly $1 billion in costs to fund those initial efforts to first find the drug and then all of those clinical trials to get it approved by the FDA. And this is why drugs cost so much these days, or today, because those drug costs need to fund all of those initial efforts and those clinical trials, because the success rate in identifying and getting a drug to market is so low. Only one out of every possible 10,000 compounds becomes a drug. Getting a drug to the market is a long and steppy process, as I mentioned, and it begins with identifying a protein that will have a desired effect on the disease of interest. And once you have validated that desired effect, you can go and find a lead compound for that protein. There are lots of different ways to find lead compounds, either through nature or maybe just designing a compound that binds to the desired protein in a certain way. And from there, you go into the optimization stage, where you will make a bunch of analogs or a bunch of libraries of different compounds based on that lead compound, and then you'll continually test them. You'll reassess where you are with each library or with each compound to try and optimize those different properties, such as the desired biological activity, but also stability in the body. And once you have found a compound that really just has the best activity in all those different effects, you can go on to take it onto phase 1, 2, and 3 trials to make sure it's safe in humans. And then you can finally submit your drug for FDA approval, and they can simply tell you that you need more data, or they can downright deny your application. Since your goal is to identify drugs that bind to hemoglobin, it's probably important that you know what hemoglobin is. This is a really important protein because it carries oxygen throughout your body, in your blood. And it is a tetrameric protein, as in it's got 4 subunits. Here's a monomeric portion of the hemoglobin protein. And how hemoglobin transports that oxygen is by having the oxygen molecules actually bind to the iron 2+ ions of hemoglobin's heme group. So here's the structure of the heme group. You can see the iron 2+ ion right there in the middle. And the oxygen will coordinate there, and that's how it gets carried throughout your body. Interestingly, you can actually remove the heme group of hemoglobin by simply treating the protein with an organic solvent at an acidic pH, We did this with acidic butanone, and this removes the heme group, resulting in the protein known as apohemoglobin, which is just hemoglobin without the heme, which means it is our hemoglobin protein with an empty active site. So really, you are trying to identify binders of the apohemoglobin protein, as in you're trying to find compounds that bind to that empty active site of hemoglobin. You're going to do this by working in teams, and each team is going to have one of these scaffolds shown. You'll then make one of the compounds within each scaffold, and you will test it for its binding affinity, its binding activity, to the empty active site of hemoglobin. You'll then compare your data with others and discern those most potent binders, the most effective drugs for the apohemoglobin protein. Now, how are we going to assess how well our drugs bind to that active site? We're going to take advantage of some previous work by Stryer, who showed that when this small fluorescent molecule, ANS, is introduced to the apohemoglobin protein, it forms a complex that is really strongly fluorescent. However, if you just make a solution of that native heme group, just make a solution of it and introduce it to this complex, that fluorescence is lost. So this implies then that ANS binds to that active site of apohemoglobin, but that it can get kicked out, as in that binding is reversible, and when it gets kicked out by a better binder, then we can see the loss of fluorescence. So instead of adding in some heme, though, we're going to kick out ANS with our drug. And so what we should get, if we follow this displacement of ANS, if we follow it via fluorescence, we should get this dose-response curve shown here. So initially, when it's just ANS and the apohemoglobin protein, we should get a strong fluorescent signal. But as we add in more of our drug to the complex, we should kick out ANS and result in the loss of fluorescence. So then we can take our dose response curves and identify the IC50 value, or the concentration at which we observe 50% inhibition, or in our case, binding. And this is the representative measure that we use to compare different drugs. Okay, so the more potent the drug, as in the better the binder, the lower the IC50. So here we can see we have drugs A, B, and C. The most potent binder, or the most effective drug, would be drug A, because its IC50 value is the lowest. And that means that we need the smallest amount of it to have the desired effect. Okay, so we just compare these drugs using their IC50 values. Alright, so that's all of the background. So again, remember that the big picture here is that there are three main parts: the synthesis, the assay, and the analysis. So this first day, you're just going to perform the synthesis of your drug, so let's get to that. All of the drugs in this experiment are very straightforward to make because they're all one-step syntheses. For the esters, this is a simple nucleophilic acyl substitution reaction between a benzyl alcohol nucleophile and your acid chloride electrophile. And the benzyl alcohol is the portion that's going to differ depending on the specific drug you're going to make. To set these up, all you have to do is add the benzyl alcohol to your round bottom flask, followed by your reaction solvent DCM. Then you're going to add your benzoyl chloride, followed by DMAP, or dimethylaminopyridine, and triethylamine. DMAP is a nice nucleophilic base, so we're going to use it in catalytic amounts, and our stoichiometric base is going to be triethylamine. You are going to need to let these stir until the next lab period, but then the workup is really straightforward. Just transfer your crude reaction mixture to a separatory funnel, rinsing your round bottom with DCM, and then you're going to want to wash your organics with 2 molar HCl 3 times. This should effectively remove your triethylamine and DMAP from your organic. Then you're going to back extract your aqueous layer with DCM 3 times. Make sure you've got all the product in that nice organic layer. And then you're going to dry the combined organics over sodium sulfate, filter, and rotovap. For all of the drugs, you will need to analyze them via proton NMR. So make sure you get a proton NMR of your product at some point during the experiment. The single-step synthesis of the chalcones is an aldol condensation reaction between the aldehyde of 4-formyl benzoic acid and the ketone of acetophenone. So it's this aromatic group that's going to differ depending on the different drugs. And you just subject these two to sodium hydroxide base, and you form this very stable alpha-beta unsaturated ketone. The stability of this conjugated system drives this reaction to completion. To set these up, you're just going to add your different acetophenone to the round bottom flask, followed by your reaction solvent, absolute ethanol. Then you just add the 4-formyl benzoic acid, followed by the sodium hydroxide base and water mixture. You only need to let these stir for an hour, and then you're going to work it up by adding 2 molar HCl to the reaction until the reaction mixture is acidic and has a pH of about 3 as tested by pH paper. This will cause the product to precipitate out, so then you just isolate it via vacuum filtration, and you're going to rinse the product with cold water. Don't forget to take your NMR, and with this one you're going to need to do that with deuterated DMSO as opposed to chloroform. The Naphthamides also utilize a nucleophilic acyl substitution reaction between the amino group, this is the more nucleophilic group of our 5-amino 1-naphthol starting material, and then the acid chloride electrophile. So now it's the acid chloride that's going to differ on the different drugs, and you just subject these two with stoichiometric base, triethylamine, and you will get your product. To set these up, just add the 5-amino 1-naphthol to the round bottom, followed by your reaction solvent DCM. Then add your acid chloride, followed by triethylamine, and you're going to let these stir until the next lab period. For the workup, you're just going to transfer the crude reaction mixture to the separatory funnel and rinse your flask with DCM, and then you're just going to wash the organics with 2 molar HCl. You're going to do this 3 times, and then you're also going to wash your organics with brine once. Then dry the organics as usual over sodium sulfate, filter, and rotavap, and don't forget to take that NMR. And finally, the conjugated esters. This one-step synthesis utilizes a Horner-Wadsworth-Emmons reaction. The sodium hydride base deprotonates the acidic proton on our triethylphosphonoacetate starting material, which renders this an awesome nucleophile that will add to our benzaldehyde electrophile. And this is the part that differs depending on the different drugs. You can see we have another alpha-beta unsaturated system here, so this is the driving force for this reaction. These are a little bit more complicated to set up. First you add your sodium hydride followed by THF, and you're going to cool the flask to 0, at which point you will then add your triethylphosphonoacetate reagent. You're going to let that warm to room temperature and stir for about 30 minutes. Finally, you're going to add your benzaldehyde, and you're only going to let that stir for an hour before you work it up. This workup you're going to transfer to the separatory funnel with diethyl ether, and then you just wash your organics with DI water. You're going to do this twice. You wash it also with bicarb 1 time and then brine 1 time. And then as usual, you're going to dry your organics over sodium thiosulfate, filter, and rotavap. And of course, don't forget that NMR. Our penultimate topic is safety. As I'm sure you have come to realize in our 370M organic lab, pretty much everything you work with is hazardous or at least a possible carcinogen, so make sure you take all the usual precautions when you're handling these reagents, including always wearing a lab coat, always wearing your glasses or your goggles, and using gloves whenever you are performing chemistry. So finally, you need to get going. This is a long experiment, and there's definitely a bottleneck at the assay step. So if you can, do your best to get your synthesis done on day 1 so you have as much time as possible for the assay and analysis. We only have 1 fluorimeter, and so while you're waiting to get on the instrument, or if you're an early user of the instrument, be sure to work on the docking tutorial. This is a great activity and will allow you to visualize how your drug is binding to that target pocket, the empty active site of hemoglobin. This will definitely help with your analysis and your write-up because you'll get to make a really nice figure that will show how your drug is binding to that target pocket. So get going and good luck! I hope your drug is the best binder of them all.